Hello everyone, welcome back to my channel. In today's video, we'll study another biomolecule, and that is fat. They are also known as lipids. So we'll see how they are formed. Now lipids or fats, they are formed by the aggregation of two molecules, fatty acids plus alcohol. Right? As the name fatty acid is there, that means the functional group here will be COOH, whereas in the alcohol it would be just OH. Now here the condensation reaction will happen and it will lead to the evolution of water molecule, and these two parts will be joined together. Means fatty acids and alcohol, they will be united together at the expense of water molecule. And in chemistry also we have seen that whenever carboxylic acid group condenses with the alcohol group, then this is known as esterification. And because of esterification, the product that is formed, it is known as ester. Okay, so this is the main logic. Fats or lipids, they are formed by the aggregation of fatty acids and alcohol. Preferably in the case of fats, the alcohol is generally glycerol. Okay. Now glycerol is very important. It's an example of trihydric alcohol. When I say trihydric alcohol, it means it has how many groups? Yes, 3 groups of alcohol. So its structure is CH2OH, CHOH and CH2OH. This structure is very important. The chemical formula is important. You can see here in the structure that 3 OH are there. Now let's move on and get a bit more knowledge about fatty acids. Fatty acids are divided into 2 categories. One, saturated fatty acids. I'm writing short FA. FA means fatty acids. And unsaturated fatty acids. Now saturated fatty acids are those which contain single bond between the carbon atoms. So ghee is an example of saturated fatty acid, which is quite common at everybody's place, and it is solid at room temperature. That's why I have given you the example of desi ghee, because ghee stays solid at the time of winters because room temperature drops down. Okay, so these have high boiling point and melting point. Fine. You have to learn 2 examples here which are being given in the NCERT. One is palmitic acid and another is arachidonic acid. Okay, we'll discuss about palmitic and arachidonic acid now. Now if you see the structure of palmitic acid, we just need to emphasize upon the number of carbon atoms in palmitic acid. Okay, so here it is 1, here they are 14, because this is whole to the power 14, and here it is 1. So overall carbon atoms in the case of palmitic acid are 16. This is important to learn. Okay, so palmitic acid falls in the category of saturated fatty acids and it has 16 carbon in its structure. Whereas if you talk about arachidonic acid, then it possesses 20 carbon in its structure. So it would be written like this. This is important to learn. Fine. Now Another thing, saturated fatty acids. Saturated fatty acids you have already understood. Let's come on to unsaturated now. Unsaturated fatty acids, they may contain double bond or they may contain triple bond in their structure, and they are liquid at room temperature. Because they have low melting point and boiling point. So we have discussed saturated and unsaturated fatty acids, and these aggregate together with the glycerol. Let's take an example whereby we will be learning how actually lipids are formed. So let's take 3 molecules of palmitic acid, okay? So if you are going to take 3 molecules of palmitic acid, you are going to write like this. Now let's make the structure of glycerol. You know, it's a trihydric alcohol, so 3 OH groups will be there, okay? They will be combining together. Now to do esterification, we would be requiring 3 molecules of palmitic acid. So I am just writing palmitic acid 3 times and we'll be forming a triglyceride. Just see here, these will be condensed and overall product we have to join. So how many molecules of water will move out? Of course 3, and the resultant product will be our tripalmitin, which is an example of a lipid or a fat. So let's write all together. Even you can practice. I hope that's easy. It is just an esterification reaction. It would be just O and this here, this CH2 here, and here we'll be writing the remaining structures of the palmitic acid. So you can see we have just taken out these 3 molecules of water out and the remaining structure is joined. Now here this is our tripalmitin. Now tripalmitin is an example of a lipid or a fat. How easy it is. Now here you can see that we have done a esterification reaction at 3 places. Fine. That's why it's an example of triglyceride. Fine. If condensation reaction happens at just 2 places, it will lead to the formation of diglyceride. Means if 2 molecules of palmitic acid will be used, then it will lead to the formation of diglyceride, because 2 molecules of carboxylic acid group, they will be getting condensed with 2 molecules of the alcohol. Okay. Now if we are just doing condensation at one place, then it will be known as monoglyceride. Which are more common? Triglycerides are more common. Now sometimes what happens is, in one of the chain, let's say this one, sometimes in this chain phosphate group is attached. Okay, phosphate group is attached. So when you are attaching phosphate to the lipids, what would be the name? Yes, the name will become phospholipids. And these phospholipids are quite common in the plasma membrane of animal cell specifically. However, lipids are also found in the plasma membrane of the plant cell. One of the important phospholipid that you have to learn is lecithin. Okay, this you have to learn. So phospholipids are derived from lipids only. When we add on phosphate group to the lipids, they will become phospholipids, and they are polar in nature, means they are hydrophilic. Now let's talk about the plasma membrane a bit. Plasma membrane, you know, it is bilayer, and whenever you have generally drawn like this. This is the way we represent the lipids in the plasma membrane. Now here I just want to make one thing clear. Generally a lipid is shown like this, and this is the polar part, and this is hydrophilic in nature, means it is water loving. Water loving means it can stay close to the water, whereas these two chains are non-polar in nature, and they are hydrophobic, means they have a phobia with water, means they are water repelling. Okay, so plasma membrane, suppose this is the animal cell, and you know that the outermost part of animal cell is the plasma membrane. So if I want to show plasma membrane, this is the zoomed view of plasma membrane. So it is a bilayer. You can see bilayer means two layers. So two layers of lipids are there. One is this one and one is this one. And you have also studied in the chapter Fundamental Unit of Life, Cell, that proteins can be found externally. Proteins can be found internally, because why am I saying internally? Because inner side cytoplasm is there, and this is outside of a cell. Okay, and some proteins, they are just crossing the membrane, crossing the bilayer, and such proteins are known as transmembrane proteins. Okay, so extrinsic will be found on the outer side, intrinsic will be found on the inner side, and transmembrane, they will be piercing, they will be embedded in the bilayer. Okay, so lipids or fats means one and the same thing. They are formed by the aggregation of fatty acids and glycerol. Esterification reaction happens. So actually the product that we receive after esterification, it is an example of an ester. So in just few seconds back we made tripalmitin, and this tripalmitin is an example of an ester. If you are doing condensation reaction at 3 places, it will result in the formation of triglyceride. If you are doing at 2 places, it will lead to the formation of diglyceride, and at 1 place will obviously result in the formation of monoglyceride. Lipids are an important component of the plasma membrane, and bilayer is composed of lipids. Lipid contains two parts, the polar part and the non-polar part. Generally, phospholipids, they show such type of polarity. Okay, so lecithin, which I have just taken as an example, falls in this category where the polar head includes the phosphate part and it is hydrophilic in nature, whereas the remaining structure is hydrophobic and it stays inside so that it can be away from the water. Okay, I hope you have understood. Don't forget to share and subscribe my channel, Online Biology Tutorials. Stay connected for more videos. Happy learning.